All right, guys, we're going to be moving forward in our cell unit, and we're actually going to be doing our second and last set of notes for this particular unit. And so this particular set of notes is going to be all about the cell membrane or the plasma membrane and how molecules are transported across it. So just a little bit of background about the plasma membrane. Some of this was mentioned in our last set of notes when we went over the cell parts and organelles. But remember that the plasma membrane is the barrier. It separates the cell from everything else. And so when we're dealing with the cell, we're going to have everything that's on the inside, which would be considered intracellular. And then we're going to have everything that's on the outside, which is going to be extracellular. And so hopefully you remember already that the cell membrane is responsible for regulating the passing of materials in and out of the cell. We know that it's what we call, hopefully, selectively or semi-permeable. Both of those terms mean the same thing. But remember that the plasma membrane is picky. Because of its unique structure, you know, it has the phospholipids and other molecules that we're about to see. But because of its unique structure, it doesn't just let anything cross the cell membrane. So because some things can and some things can't, we call it a selectively or semi-permeable membrane. So that's its main job, is to kind of regulate the in and out of molecules and substances in and out of the cell. But it's not its only function. There's actually some other things like communication. As we go through the different components, we'll see some of these other functions come to play. Things like cell recognition. It can also be, things like transport as well. So the cell membrane structure is a pretty unique organelle or part. It's got four main components, and we kind of describe our plasma membrane as with the fluid mosaic model. Fluid because it's kind of a flexible. It's not rigid. It can actually kind of be changeable, if that makes sense. Like these phospholipids can move around. So it's not like a rigid, non-changing structure, okay? Dynamic, that's the word I was trying to think of. And so the fluid mosaic model describes the fact that it is kind of flexible and changing, but it also has multiple components, and that's where you get that mosaic portion. So there's four main components of the cell membrane, the main one being the phospholipids, but we're also going to include cholesterol, proteins, and carbohydrates. So of all of our macromolecules that we talked about, 3 of the 4 macromolecules can be found in our cell membrane. And so here's another example of what it could look like. So we've got multiple components. We've got our phospholipids, we have big chunky proteins, we have carbohydrates, we have cholesterol molecules. And so the reason it's kind of a fluid model is all of these little phospholipids, they can shift around and change position. They aren't like tightly bonded to each other, and that's where you kind of get that dynamic aspect of our membrane. Now they can't jump across from one side to the other, but within the same side, they can kind of move around and shift and change places. So we're going to go through each component. We're going to start with the phospholipids, and the phospholipids are the main component. So the majority of the membrane are going to be these amphipathic phospholipids. And what's unique about the phospholipids is that they have both a hydrophilic region and a hydrophobic region. We actually looked at this back when we did lipids, but the term that describes that is amphipathic, so it kind of has two natures. We have the water-loving head and the water-fearing tails. Now, something you have to understand about cells is that cells are aqueous. There's always going to be a solution inside of the cell. You know, the cytoplasm has lots of water in it, along with other things that are dissolved. But then outside the cell, you're also going to have some type of environment that has water. Now, you might be thinking, well, my skin. Obviously, we're not soaking in water right now, but the skin cells on the outside are dead. So living cells are usually always going to be bathed in fluids. And so you have to understand that there's always going to be water inside the cell, and there's always going to be water outside the cell. And so what happens is, because of the structure of these phospholipids, they naturally turn so that the tails are away from water. And what that gives us is a bilayer. The basic structure of our cell membrane is a phospholipid bilayer or 2 layers. And so because the heads like water, they're fine touching, and then the tails don't like water, so they turn inward. And so that's why you have these heads touching the water outside of the cell, and then you have heads touching the water inside of the cell. And that is going to be your basic structure of your membrane. Now there are 3 other components that we're going to talk about. The next one is cholesterol, which is another type of lipid. Cholesterol is actually a steroid, and steroids have a little bit different of a structure, but like all lipids, they are hydrophobic. Okay, they do not like water. And so I want you to notice where you find these cholesterol molecules. They are down here amongst the hydrophobic tails. Okay, they are also going to be in the area that does not like water. And so they're going to be within those tails, and what they do is they kind of stiffen or stabilize. They're going to give that membrane some extra structure, keep it from being too fluid, because we do need it to have some stable structure to it. It also decreases the water solubility, so that way that plasma membrane is less likely to dissolve in solutions. Another major component of our cell membrane is going to be proteins. And hopefully you remember that proteins always have a very specific shape, and they always have a very specific function. And so we have several different types of proteins. In general, you could find proteins that are integral, which means they go all the way through from top to bottom. Okay, so this over here is an integral protein. These are integral proteins. Or they could be peripheral. And if you think about your side vision, that's your peripheral vision, your peripheral proteins are going to be on the side. So these are ones that don't necessarily go all the way through, inside to outside. They're just going to be on one side of the membrane. Now, the functions can vary when it comes to your proteins. Some of them are going to be used for transport. They could be channel proteins, they could be carrier proteins, or they could be pumps. And we're going to take a look at some of those specific proteins when we get to transport. They could be protein receptors. As we move forward in biology, there are several times where we talk about proteins acting as receptors. So proteins can bind things like hormones or neurotransmitters or medicine, but these proteins are going to receive chemical signals, and by doing that, they can play a role in cell communication. They can also be a part of the cell identification. Like your ABO blood group, the reason you're type A or the reason you're type O has to do with the proteins that you have or don't have on your cell membranes. And then they can also play a role in connections. So there's different cell junctions that hold cells together, and many of those involve protein structures. So here's just another example of all the different types. Some proteins could be enzymes, and they could catalyze chemical reactions. We haven't talked about enzymes in a whole lot yet, but we will continue to talk about them in our next unit. Several of these are allowing molecules to pass through, and then some of those peripheral proteins could be things like your receptors. All right, the last component of our cell membrane are going to be carbohydrates, and hopefully you remember that carbohydrates are sugars. Now, these are not necessarily sugars that give us energy, okay? Sugars do give us energy, but that is not the function of these carbohydrates. They're actually short chains. They're kind of between a disaccharide and a polysaccharide. A lot of times they're going to be like maybe 10 to 20 monosaccharides in length. So they're going to be shorter chains, not these huge polysaccharides. But typically you find these sugars in the extracellular matrix, which means they're going to be in the components outside of the cell. And they're either going to be attached to a protein, and if that's the case, they're called a glycoprotein, like this one here. Or they could be attached to a lipid, and if that's the case, they're called a glycolipid. And so glyco refers to the carb, and then the other term refers to what they're attached to. But the purpose of the carbs in the cell membrane is identification. So between proteins and carbohydrates, those play a major role in cells recognizing each other and knowing whether something is part of you or not part of you. They kind of serve as a name tag for what kind of cell that it is. So going back to the function of our cell membrane, remember that the main function is that it's going to be a selective permeable barrier that's regulating what enters and leaves the cell. And what determines that permeability has to do with several factors. It could be based on size. It could be based on what kind of molecule it is. Is it polar or nonpolar? Is it hydrophilic or hydrophobic? Does it have a charge or is it uncharged? And then obviously the concentration is going to be important in determining the movement across a membrane. And so here's just a kind of an overview. So if they're small and hydrophobic, notice they easily cross the membrane, because remember that they have to get through these tails, okay? If they're uncharged and polar, some of them can easily cross, some of them not so much. If they're large, typically, if they're going to cross, notice they have to have proteins. They can't do it by themselves. And then ions, same thing. They can't easily cross that membrane, and so they're going to have to also require some proteins. And so, kind of like what that little graphic just showed you, if they're small and uncharged, they typically move across easily. So gases like oxygen and carbon dioxide, vitamins, steroids, small things that are hydrophobic normally move across. And what's going to drive that movement is a concentration gradient. Now, I don't have this defined here, but I do want to talk about it because it's going to be present throughout the rest of our lesson. But a concentration gradient is a difference, okay? The gradient means there is a difference in concentration inside and outside the cell, okay? And the larger the difference, the larger the gradient, okay? So a gradient is just simply a difference in concentration in and outside of the cell. Now, water is another one. Now, water can easily cross, but we've already talked about those little fatty acid tails. They don't like water. So water is not going to be able to easily pass through the bilayer itself. So luckily we have these proteins called aquaporins. They are specific to water, but they're proteins that allow water to cross the membrane, and so water can typically always easily pass through. Now your larger molecules, because of size, they can't easily move through, or charged particles can't easily move through. And so typically these are going to require some type of assistance in moving across. So things that are big may require proteins, either channel or carrier proteins, or they may be brought in a vesicle through processes called endocytosis, or they could be released through the membrane by a vesicle in a process called exocytosis. And so these are the kind of things that we're going to be looking at through the rest of the notes. So in general, there are two main types of transport. And to me, these are pretty easy to remember because the words kind of give it away. We either have passive transport or we have active transport. And hopefully you know that if you want to be active, you have to have energy, okay? So really one of the main differences between the two is whether or not they require energy. And then whether or not they require energy is based on the direction that molecules are moving. Are they moving with or down the concentration gradient? Or are they moving against or up the concentration gradient? So I want you to think of this as kind of like uphill or downhill. Now, in passive, they're moving from high concentration to low concentration. Molecules have a natural tendency to spread out, okay? So anytime they're moving from high to low, that's kind of like moving downhill. You don't need energy to do that. And we can say that that is down the gradient, or that is with the gradient. So kind of think about a river. If you're going with the flow of the river, you don't really have to do anything. You just float and the river's going to carry you. That's the way these molecules move. If we have a gradient, they're going to naturally want to move from high to low. Now, active on the other hand is going to be like going uphill. We're trying to move molecules from where there are less of them to where there are more of them. And molecules do not naturally move that way. So this is like going upstream in the river. You're going to have to put in some work. You're going against the flow. And so active transport is going to require energy, typically in the form of ATP, because we're going against or up the gradient. And so now we're going to take a look at some specific examples of each one. So for passive, there's three main types. We have simple diffusion, facilitated diffusion, and osmosis. Now in simple diffusion, this is just a basic high to low, okay? It's diffusion, it's passive, all of these are going to be high to low. But the key to simple diffusion is that it's going directly across that lipid bilayer. So these would be some of those small hydrophobic uncharged molecules like gases, okay? They're going to easily just pass through the membrane, and as long as there's a difference, they're going to naturally flow from high to low. Now, the next type is facilitated diffusion. Okay, we're still in passive, so once again, we're still going to see that high to low. Energy is not going to be required, but to facilitate means to help. So in this case, this is going to be where you have those molecules that can't really move directly across the bilayer. Instead, they're going to have to rely on one of those proteins. So the key to facilitated diffusion is that it's a high to low movement with the help, remember facilitated means to help, with the help of a protein. So in pictures, you can see that the molecules are passing through these big chunky proteins. If it were simple diffusion, they're just passing through this bilayer here, okay? So we're still moving down the gradient. We're just moving down the gradient through a protein. And this is going to include maybe some polar molecules or some a little bit bigger substances or maybe charged particles, things like glucose, amino acids, or some ions. And then we have osmosis. Now, osmosis technically is the movement of water or the diffusion of water across a membrane. We mentioned earlier that water movement does involve a protein, so this is also kind of an example of facilitated diffusion. But the thing with osmosis is water is always going to move, okay? Because water can freely move across the membrane, anytime there is not a balance in concentration, anytime we do have that concentration gradient, water is going to move from where there is more water to where there is less water. Okay, so we're still moving from high to low, which is why osmosis is passive, but it's specifically going to be the movement of water. Now, in order to understand osmosis, because it's kind of a big deal for cells, this is one of those homeostatic mechanisms. We have to maintain water balance. So to kind of fully understand osmosis, we have to understand tonicity, which is the concentration of solute. Okay, this is important. The concentration of solutes dissolved in that water, and then how much that is going to affect their diffusion. Okay, so you need to understand that if there's more solute, there is less water, because water is our solvent, right? If there is less solute, there is more water. So when we use osmosis, or when we talk about osmosis, there are always going to be two solutions, okay? We don't really use these terms that we're about to talk about unless there are two solutions. So in terms of biology and in terms of cells, our two solutions are always going to be inside the cell, that intracellular solution, and it's going to be outside of the cell. And so anytime we're talking about isotonic, hypotonic, or hypertonic, it's always comparing two solutions, inside the cell and outside the cell. So the word isotonic, iso means equal. Isotonic solutions are those that have the same amount of solute. Their concentrations are the same. Hypotonic, hypo means low. These are solutions that have less solute and therefore more water. And then hypertonic, hyper means high. These are solutions with a higher solute concentration. So remember, we're talking about osmosis though, so we're talking about the movement of water. So isotonic is going to be equal, hypotonic is less solute and more water, and hypertonic is high solute and less water. So just kind of a little bit of practice here. You don't really have anything in your notes that you need to do, but let's just take a look at these three examples. It says describe the solutions outside of the cell. So these are my cells, these little bag-looking structures. So each cell has a concentration of 10% salt, okay, that means 90% water. And so these represent the solutions that are outside of the cell, okay. So we have a 10% solution in the cell, and we have a 10% solution outside of the cell. Beaker A would be isotonic, okay. Now here we have a 10% salt solution inside the cell, a 2% salt solution outside of the cell. So we're talking about the beakers here. So this beaker would have less solute than the cell, and so this would be a hypotonic. And then over here we have a 30% compared to a 10%. 30 would be more. So this beaker would have a hypertonic description because it has higher solute. So just a little practice there. You have to understand, once again, that there's always two solutions. So if the beaker is hypotonic, then the cell would be hyper. These two words always go together. You can't have one without the other, okay? All right, so in general, the more solute there is, what happens is water kind attaches to those solutes, it binds to them, and then it can't easily freely move around. And so the more solute there is, the more water is going to go there, okay? Water is always going to move to solute. So a couple of things that you can hopefully remember is water always moves from hypo to hyper. It's always going to move from the area that is hypotonic, because there's more water there, to the area that is hypertonic, because there's less water there. Something else you can remember is that solute sucks or hyper sucks. Water is always going to move towards solute. This is why when you eat salty foods, you get thirsty, or you aren't supposed to drink salt water, because salt sucks, solute sucks. It's going to suck the water out of your cells. And so when we're talking about living cells, okay, it's important that living cells, especially animal cells, maintain isotonic conditions. Now, when conditions are isotonic, there is no net movement. Okay, now net means it's not in a particular direction. It doesn't mean there is no movement. Even in isotonic conditions, water molecules move, but they're just moving in and out equally. Okay, so that means they're not moving in one particular direction. So that's what it means by no net movement. Okay, so in isotonic conditions, the cell is pretty much going to be about the same, and this is what we want. Okay, so in terms of water balance, we want isotonic conditions. If you place a cell in a hypotonic solution, water is going to enter the cell. And if you place a cell in a hypertonic solution, water is going to leave the cell. Remember that hyper sucks. Water is going to move toward the solute. So hyper has more solute, water is going to go toward it. And so this is why we have to be careful about water balance, because we don't want our cells to either be gaining water or losing water. Now, it is slightly different in plant and animal cells, so we're going to take a look at these individually. When it comes to animal cells, isotonic is good, cells are normal. Okay, isotonic is water balance. Now, hypertonic, like we said, hyper sucks. So what happens is that water is going to flow out of the cell, and it can cause your cell to either shrivel up, or the word for that is crenate. Okay, crenate and shriveling is the same thing. So this is what your little blood cells would look like, or your little cells would look like. That doesn't probably sound like a good thing. Okay, if our cells were in a hypotonic solution, water is going to go into the cell, and so the cells are going to swell up, and sometimes they could swell to the point that they could actually lyse or burst. Okay, and so we would not really want that either. Okay, we don't want our cells to be swelling or shriveling. Neither one of those is going to be good. So here's some pictures of actual blood cells under the microscope in various conditions there. So water balance is essential in terms of us as humans. Okay, this is why dehydration is so serious, or you could also have overhydration. But we have to make sure we maintain water balance. If you ever paid attention to the saline drips, the natural salinity in our blood and body fluids is 0.9% saline. So it's very important that they give you that in the bag. You would not want them to give you distilled water or something that had more salt, because once again, we don't want our cells shriveling or swelling. Now, it is definitely a little bit different in plants because of their cell wall. So remember that animal cells don't have a cell wall, plant cells do. And so where our cells, I mean, the whole concept of iso, hypo, and hyper are still the same, but what happens to the cell may be slightly different. So in isotonic conditions, what happens is the plant is technically fine, but it's not completely filled up with water. Okay, so there's water in there, water's moving in and out equally, but the cells actually lose a little bit of their structure. Okay, if that vacuole is not filled up, then the cell is flaccid or limp. So it's not hurt, it's just not really filled up with water. And so for a plant, that's, you know, they like to be big and perky, right? And so this is going to be when the plant looks a little limp. Okay, all you have to do is add water and they'll perk right back up. Now, in hypertonic solutions, just like with animals, it's going to cause water to leave the cell. And what happens in a plant cell is as the water leaves, it actually causes the cell membrane, in this picture here or in this picture here, when that water leaves, it causes that cell membrane to kind of shrivel up and pull away from the wall. And that's a process called plasmolysis, or you can say that it plasmolyzes. That would not be good for a plant, okay? They're going to look more than limp. In fact, they may die if they start to plasmolyze. And then hypotonic is actually not all bad for plants. Because they have the cell wall, it won't let water just keep going in and keep going in and keep going in until it bursts like an animal cell. Instead, what happens is the water goes in, it fills up the vacuole, and it causes the cell to become turgid. And what happens is when the water fills up, that water pushes against the cell wall in something known as turgor pressure. And that's actually part of the plant cell structure, okay? So we actually kind of want a full vacuole in the plant cell, and it won't overfill and burst like an animal cell does. So for plants, hypotonic isn't too terribly bad because it actually causes their cells to be full and happy, okay? All right, so that takes care of all of our passive transport. Once again, passive transport is all high to low, no energy required, but sometimes we need to move molecules in the opposite direction. So sometimes we need to move molecules against the concentration gradient, or we need to move them from low to high. And in order to do that, just like when you're going uphill, it's going to require some energy. Now the proteins are going to be involved in here as well. Typically when you're talking about a protein that's active, we call them a pump. So anytime you see the word pump, you automatically know that it is going to be active. And a lot of times in the picture, you're going to see that energy molecule. If all else fails, you can always count and look at the concentration, okay? So we do have a few examples here of active transport. One is the sodium-potassium pump. Now, you probably haven't learned about this too terribly much in school, but in terms of like anatomy, like what I teach over here at the college, when we cover neurons and we cover like nerve impulses and we cover muscle contraction, the sodium-potassium pump is kind of a big deal, okay? It's found in the membrane of animal cells. It's very, very important in terms of like muscle contraction and nerve impulses, so it's kind of a big deal. But all you really have to know for now is that it is a pump, and therefore it must be active. And so the way the sodium-potassium pump works is it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell, and in both cases they're moving against their gradient. So it's going to require energy. You can see ATP in the pictures down here, and that's how you know that it's active, okay? Another example that is part of like cellular respiration and photosynthesis is something called a proton pump. And what it does is it pumps hydrogen ions across the membrane and it concentrates them. And anytime you concentrate ions, it stores energy. So it creates potential energy. Because remember, they want to go high to low. So when you make them go to one side, it creates a lot of stored energy there. And then a third type or a third example of active transport is something called bulk transport. You know, sometimes things are just too big and they're never going to be able to move across the membrane. So instead what we do is we use the membrane to either bring these big molecules in or expel these big molecules out. And we do it using these little membrane sacs called vesicles. And so we're talking big molecules like food particles. It could be something like another cell, like a bacteria, but bulk transport, because it involves the membrane kind of surrounding and fusing in different ways, it's also going to require energy. And so this vesicular transport, or using vesicles, it can bring things into the cell by a process called endocytosis. And this is where the membrane literally kind of surrounds or engulfs these substances, and then it kind of pulls it in and forms that vesicle. Now there's actually 2 main types of endocytosis. There's phago and there's pino. Now phago is food, okay? Phagocytosis is when the cell is bringing in large particles, usually for consumption, kind of like cell eating. And then pinocytosis is when it's bringing in bulk fluid. So this is kind of like cell drinking. And then exocytosis is just going to be the opposite. This is when the cell is expelling or releasing contents. So the vesicle will fuse with the membrane and release the contents to the outside of the cell. And so here you have some pictures that kind of represent your endo and exocytosis. So once again, endo is bringing in a vesicle, exo is expelling out from a vesicle. Here's phagocytosis, bringing in a little food particle. Many of your white blood cells are what we call phagocytes. And what they do is they go around and eat foreign particles like bacteria or microorganisms, and they do it using a process called phagocytosis. Pinocytosis, once again, is bringing in fluids. Here you have an example of a cell engulfing a pathogen, or you could have an amoeba engulfing food. And so all of these are just some pictures of what that would look like. All right, and finally, you have this little chart here. It may not have every piece of information, and it's not in your notes, so it may be something you want to go back to in the PowerPoint on Google Classroom, or you can maybe make a copy of it in your notes at a later date. But this is just kind of an overall review. So remember, we had 3 types of passive transport. Passive is always high to low with no energy. We have basic diffusion across the membrane, facilitated diffusion through a protein, or osmosis, which is the movement of water. And then active transport, of course, required energy. That could be those protein pumps that are pumping ions, or it could be endo or exocytosis, those bulk transports. So that is going to take care of the notes for today. You're going to spend the next couple of days doing some labs to help practice and be able to see and answer some questions about different types of transport. And I hope you guys are doing well and everything's going smoothly for you. If I haven't seen you in a while, hopefully I will see you soon, and you guys have a great day.